Okay, so I have one disclosure, and that is the fact that I used to consult for Salix Pharmaceuticals, and this reminds me to thank them because Salix Pharmaceuticals are a major donor for our event today, which I forgot to mention this morning. Okay, so Jack mentioned a word called dysbiosis, and this is defined as an unhealthy change in the normal microbial ecology of the gut, okay? So you have your normal ecology of the gut, your bacteria are living in balance together, and suddenly you have this change. And this change has been associated with many different diseases, some of them quite obvious, like inflammatory bowel diseases, because they happen in the gut. And then you've got others, like what John talks earlier on, or yesterday, about affecting your mood. Now, Jack mentioned that he goes on tangents, and I would argue that I am nothing but a tangent. So I remember John asked me to come over to Cork and give a talk. And I was just about to go up on stage and give a talk, and he says, did you use the term dysbiosis in your talk? I was like, why? He's like, well, the chair of the department hates that word. I was like, John, it's on every slide. Why didn't you tell me like 5 minutes ago? Anyway, the reason why he hates it, Professor Fergus Shanahan, right, John? Yeah, is because it implies causality. Okay, so basically a dysbiosis, if you say that, it's going to be like, okay, well, these associations are real, they're causing the disease, but they're not necessarily doing that. A change is a change, and you have to prove that that change is indeed unhealthy and causing the disease, and that's why we need to know whether it's a cause or a consequence of the disease. And this is really important because in the last 10 to 12 years, we've seen an explosion in microbiome research with the number of publications like just shooting up. And I've been involved in these association studies, and they're very important as a beginning to proving causality. But it's really important that we start working towards starting with associations and then working more towards mechanisms. Now, Dr. Matur spoke about this yesterday, but as a traditional microbiologist, I began my training as a bacteriologist in Trinity College Dublin. We always worked with Koch's postulates. And Koch's postulates are a series of criteria that you need to fulfill to determine whether a microbe is causing the disease or not. And again, Dr. Matur talked about this guy yesterday. And this is Barry Marshall. And I did my PhD under investigating a microbe called Helicobacter pylori. And it's a brilliant, brilliant, interesting microbe. And basically, Barry Marshall, he's from Australia, and he's finding all these, in biopsies, he's finding gastric biopsies, he's finding these histological sections. There's microbes that are associated with the mucosa. But there's so much money put into industry for antacids to treat ulcers that no one would believe him that this microbe was causing disease. So he tried to culture it, and he couldn't. And he tried to culture it again, and he couldn't. And then, being Australian, and no offense, because it's great to take holidays, he went away on a long weekend and forgot that he left his plates in the incubator. And then he came back after the long weekend, and then he saw the microbes growing. So it's good to take a break, all right? Because he actually cultured Helicobacter pylori. It needed a week to grow, because they're very fastidious and slow-growing organisms. So he was like, yes, okay, I've proven it. I've grown these microorganisms, I've classified it, but yet again, nobody would believe him. And Dr. Matur talked about this, that he took this microbe, grew it, and drank it, okay? Now, he showed that that microbe caused gastritis. He took antibiotics, got rid of it, and showed that the gastritis went away as well, fulfilling Koch's postulates. And, okay, that was good for him. When my students is in the audience, I don't expect you to do work like this, okay? I actually had lunch with Barry Marshall, and he is just as exciting as you think he would be. But we don't have that ability for a complex microbial community. It's not one bug causing the disease. maybe it's a couple of bugs, maybe it's, as Jack was talking about, it's the entire ecosystem that you have to consider. So this is a really interesting article that came out a long time ago. A lot of you won't be able to find it because you spell skepticism with a K, okay? So just write down, it's a C, all right? And basically, this article, it seems a little bit damning. It's a little bit old now at this stage, but it seems a little bit damning for microbiome research, but it's not. It gives you, you know, time to pause and think about how you're interpreting things. Think about how you're determining causality and associations. So one of the points that I've made there is, what's the functional impact of intestinal microbiotas on the host? So a way to do this, and I should have had this in the questions, but does anybody remember John's talk yesterday, who Eli was? Good, okay. We don't have that on the post-test question, unfortunately. But Mechnikov basically had a few crazy ideas, as John had said. He basically thought that microbes were slowly poisoning us and killing us. And again, yes, he did have a few crazy ideas. One of them was that we should all remove our colon and we'll live forever, okay? But then you have Louis Pasteur, who basically said that microbes are really important to life and we can't exist without them. Now, he was kind of right, but not really, because in 1946, the first germ-free animal was made. So this is exactly what it sounds like. You're growing an animal in a sterile bubble. And this comes down to a field called notobiotics. Greek for notos and bios, known life. So you're essentially controlling the microbial environment of an animal. And this can be you can have notobiotic rats or pigs. But we tend to, at UNC Chapel Hill, work with mice. They're a little bit smaller and easier to manage. But you can appreciate, as Dr. Matur said yesterday as well, that these mice are sick. If you look at the intestinal physiology of the germ-free mouse, what's circled there in red is the cecum, and you can see that it's tiny in the SPF mouse with a normal microbiota, but the cecum is huge and it's enlarged, and it is obviously different. So although Pasteur wasn't exactly right, he kind of was, because we're not normal without our microbes. Now you can have in notobiotic animals anywhere from germ-free, or you can just put in one microbe and have it mono-associated, which isn't a natural occurrence because we live with like plenty of microbes. But you can also dual associate or put in a consortium of microbes, of unknown, uncultured microbes. And this is a very powerful technique to address what we can't with Koch's postulates, to look at the effect of a complex microbial community on the host. So there are many different things in the environment that we encounter every day that can influence the composition or the function or the activity of your microbes. And this is anywhere from antibiotics, probiotics, your mood, drugs, drinking, which maybe some people did last night. I know I did. But also a huge driver of this is nutrition. And indeed, Peter Turnbaugh did a study not so long ago. It was with a very small number of patients, or a number of participants. It was 10 people. And he basically radically switched their diet, going from a very hardcore vegetarian diet to a carnivore diet. And in fact, if you read the methods in detail of that paper, one of the participants was a vegetarian and fully went onto a carnivore diet. I don't know how he convinced her to do it, or him, or whatever, but they did it. But within 24 hours, the composition of the intestinal microbiota changed. So that's a very fast shift in the microbiota. But what I'm interested in is what happens if you cut off your nutrients that you're providing to your microbes. And this brings us to a psychiatric disorder called anorexia nervosa, which I'm sure a lot of you have heard about. It's a severe psychiatric disorder characterized by extreme weight dysregulation and high rates of comorbid anxiety. And interestingly, it affects nearly 1% of women in the United States and 0.3% of men in the United States. So this is not a disease that's associated with a particular sex. Now, I always find this really interesting. We have ways to treat patients with anorexia nervosa. Okay, there's established pipelines. However, it has the highest mortality rate of any psychiatric illness in the United States. So that tells you a lot. We have ways of treating it, yet there's the highest mortality rate. So we need new, innovative ways to treat these patients. There is definitely some genetic components to the disease. And in fact, there's a GWAS study that was carried out by one of my mentors, has shown that there are specific loci in the human genome that are associated with anorexia nervosa. So if we think about the dominant traits of anorexia nervosa, it's weight dysregulation and altered behavior, such as anxiety and stress. And from what we heard yesterday and what we're hearing today, is these traits are influenced by the intestinal microbiota. So I'm really interested, based on this knowledge, knowing what impact it has on anorexia nervosa, if indeed the intestinal microbiota plays a role. And so this goes back to a couple of studies that were done many years ago, where Jeff Gordon's lab took 2 groups of mice, fed one a normal diet and one a Western diet. Can anybody guess what a Western diet is? It's high in vegetables, high in sugar, high in fats. And guess what? Lo and behold, the mice on the Western diet had increased adiposity. Now, what was innovative about this study was he took germ-free mice and colonized these mice with the fecal microbiotas from those mouse groups. Now, what's really interesting is they ate the same food, they ate the same amount of food, but the mice that received the fecal microbiota from the obese, or from the Western diet group, had increased adiposity. This was also translated to a human situation where they took twins discordant for obesity. So these are samples coming from humans now at this stage. And again, they colonized these mice with twins that were discordant for obesity. And again, the mice ate the same amount of food, the same quality of food, same type of food, and yet the mice that received microbiotas from the obese twins had increased adiposity. So this was the first study to suggest that an obesity-associated phenotype could be transmitted via enteric microbial communities. And it led to the energy extraction hypothesis. So this is it's kind of a controversial hypothesis, but I really like it. Anyway, so I like to call it the cornflake hypothesis. So if you have an obese person and a lean person, and they eat one cornflake each, actually it's 2, okay? They eat 2 cornflakes each. The microbiota from the obese person is able to metabolize the cornflake, release more energy, and then the host can absorb that energy, leading to increased adiposity. This is yet to be proven, and again, the association between the intestinal microbiota and obesity remains controversial. Now, we heard from Mary and John yesterday, and Jack reiterated it this morning, about the relationship between microbes and behavior. I thought I fixed these. Anyway, it doesn't matter. When you have a germ-free mouse, okay, again, it's not well. We know the physiology of the gut is different. But what's really interesting is a germ-free mouse is a little more chilled. It has less anxiety, okay? Again, Jack mentioned that the technology to use mice as models of human behavior are not great, but there are some models that we do use, and these mice are less anxious, but they have a higher stress response, okay? When you take these mice and give them a microbiota, an SPF, as it's referred to here, a specific pathogen-free intestinal microbiota, you change the stress response, okay? It goes down. But if you use monoassociation to colonize different, to colonize these mice, the stress response is different, okay? So you have, if you think about the gut microbiota as being a complex microbial community with lots of different microbes, and they all have different effects on your behavior, it's like a battle between microbes that are trying to make you stressed, microbes that are trying to make you a little less stressed, okay? So that's very interesting. And another interesting point about this study was there was a window where this could happen. When the mice were relatively young, you could change their mood response by colonizing them. But in older mice, you couldn't do it. So it could be that the effect microbes have on your mood is you can target microbes to change your mood when you're younger, but when you're older, like me, you're just out of luck. Okay. All right. So based on these studies and this information that we had, my team came up with an idea that these traits are that are associated with the anorexia nervosa are also associated with the intestinal microbiota. So the idea is we don't believe that the intestinal microbiota is causing anorexia nervosa. But if you are depriving this complex ecosystem of nutrients, you're going to impact it, and then that can impact the host. And if it impacts the host behavior and increases your anxiety, then maybe you're going to be more anxious and you don't want to eat. And then you have this cyclical fashion where you're not eating, and then you're again depriving your microbes of food, and it happens over and over again. So we decided to investigate this, and we did this with a prospective study. And this was carried out by a student in my labs, Susan Kleiman. And one of my mentors, Cindy Bullock, has established the UNC Center of Excellence for Eating Disorders in UNC, Chapel Hill. So what happens here is we get patients who are very, very sick, and they have to be admitted for clinical refeeding. And we call that admission T1. They are clinically refed over time until they're at least 85% of their ideal body weight. Usually when they come in, they're less than 75% of their ideal body weight. So we decided to collect fecal samples at these different time points and healthy controls and see if indeed there was a difference between the microbiotas. Now, there wasn't a huge difference between T1 and T2, before admission and discharge, but we did see that the diversity in AM patients was much lower than that of healthy controls. So perhaps depriving your microbiota of nutrients is indeed having an impact on your intestinal microbiota. So one of the interesting things is that Susan did was she collected psychological measurements when patients came into the clinic, and she found that the diversity of your microbiota was associated with your depression and anxiety levels. So the less diverse your microbiota was, the higher your anxiety and depression levels were. So this was really, really interesting. Now, if you can see in the middle graph there, the point between T1 and T2, so the diversity measures, this has got me thinking about this hysteriosis, if that's what I'm saying it right. Perhaps it looks to me like the microbes have not fully recovered at discharge. So we've pushed our microbiota so far that even during this clinical refeeding, they haven't recovered. So we're sending patients back out into an environment without a fully healthy microbiota. So our hypothesis after seeing this was that intestinal microbiotas that arise from prolonged starvation contribute to altered adiposity and anxiety levels in patients with anorexia nervosa. So what we're doing right now, and I'm going to share some of the data with you, but we're not finished this study yet. We're 4 years into a 5-year project. We are collecting 100 stool samples from well-characterized AN patients that come into the seed at T1 and T2. Now, we're also doing DEXAs on them to get body weight and a more accurate measure of body weight. And we're also collecting sex-matched healthy controls. And what we want to do is find whether there's a correlation between specific microbes or a community of microbes, multiple microbes, and weight gain over time in these patients. And we wonder, can you predict if there's a baseline level of microbes and you can, that can predict whether you're going to put on weight more efficiently or not so in these patients? That would be very, very useful to know. So as I said, we're only starting, we're 4 years into the 5-year project. We're nearly ready to start analyzing the data, but I'll just show you kind of what we're doing. If you get a patient at admission, we can give them a DEXA and you have their body weight, and yes, T2, they've been in for 16 days and put on a significant amount of weight. And you can also see that the percentage in fat has gone up over time as well. And what's really interesting about DEXA is you can isolate specific areas of the body and look at the adiposity in those areas. So we, one of the really difficult issues with AN patients is when you clinically refeed someone, you tend to put on trunk fat, which is not really good for this patient population because that's not where they want to go. You want to put it on more holistically and healthy all over your body. So we wonder, can we find specific microbes that are associated with deposition of adiposity in different parts of the body? So we talked a little bit about germ-free mice, and we want to know if there's a functional influence of the gut microbiota from these patients on the host. So the beauty of gnotobiotics is you're taking microbial communities out of this really noisy environment and then putting them into an environment where it's more controlled and you can test the functional influence of those microbial communities on the host. So similarly to the work done by Jeff Gordon and others all those years ago, we're taking germ-free mice, we're transplanting admission, discharge microbiotas and healthy control microbiotas into mice, and we're characterizing the microbiota. We're looking at adiposity and we're looking at behavior. And I'm going to show you some of the results right now. You can see in blue and red, they're the donors and the slurries we made to colonize the mice, and the mice are there in pink, I believe. I'm a little bit colorblind. So you can see that the microbes that went into the mice are distinct from the humans, all right? And what we can see is that the healthy controls right here tend to segregate from the AN patients. One thing I find really interesting here is the T1 and T2 AN samples cluster together based on the individual they came from. So there's no signature saying it's an AN or AN at admission or AN discharge. And it looks like the microbiotas, at least when they're transplanted into germ-free mice, are really, really similar. There's no distinction from them. So again, I'm starting to believe that even though you're ramping up the calories in these patients and they're putting on so much weight, their microbiotas are not changing, which is really, really interesting. And also we can look at we can mirror what we're doing in humans, doing DEXAs, we can do that in mice. And here's the first preliminary results. Now I'm going to show you preliminary and actual results and how different they are. Okay, so this was what spurred us to do it. We first transplanted microbiotas into germ-free mice. And if you look at the T1, the red line here, so we're looking at the change in weight at baseline, and it looks like mice that had microbiotas from admission, right, an AN patient at admission, didn't increase much in weight. But if you look at the second graph, they increased a lot in adiposity, so fat mass. So they didn't gain a lot of weight, but they gained a lot of fat. Then if you take the T2 situation, that did gain a lot of weight, but didn't gain a lot of fat mass. So that's brilliant. That's exactly what everybody wants. you can increase lean weight, but not fat weight. So that led us to this hypothesis that it's been known that an obese microbiota may, if we go back to our energy extraction hypothesis, that this could also happen in AN patients, right? That the T1, the acute baseline microbiota, is also capable of extracting more energies from the gut in the same manner as an obese microbiota. Now, these are two graphs which are the result of 4 years of work. 2 graphs, 4 years, lots of sweat and tears, lots of tears, especially when we saw this. You can see there's absolutely no difference, right? It doesn't matter what happens, we put in so like we were so excited about our preliminary results, but these are our actual results. 4 years of putting AN microbiotas into germ-free mice, T1, T2 healthy controls, they're all the same. The weight doesn't change, there's no significant difference, there's no difference in fat mass. So it's great to tell your graduate student that. She was very happy. So one of the, I'm kind of optimistic, all right? A lot of people will tell you that when they talk to me. So I have a very talented person I collaborate with. Her name's Fernaz Fouladi, and she got really interested in looking at what was in the donor and what made it to the actual mice. And this could be a reason why we're not seeing any impact on host adiposity. And this is a little bit difficult to explain, but basically these are all the phyla over time. We looked at, we took fecal samples over a period of a month from these mice. And basically what they found is in all of the animals, now we're not just looking at AN, we're looking at healthy NAN. Basically what we're seeing is there's certain phyla that are making the jump from donor to mouse quite well, and then the rest of them are not. And that's the microbes that make the jump belong to the Bacteroidetes phylum. Firmicutes don't make it, are much lower. So let's say, for instance, there's the microbes we think that are associated with adiposity in AN patients belong to firmicutes. Well, we're not going to be able to see it in this particular model. And this is work we just recently submitted, and I'm hopeful that it gets a good review. Now, more bad news. A Japanese study just came out showing that what we attempted to do, they actually did, right? Here is a graph showing that AN microbiotas from Japanese patients put into germ-free mice do in fact impact weight gain in their mice. I have read this paper, every inch of it over and over again. They did things very differently than us, and that's all I'm going to say about it. I'll talk to you later about it. Okay, so where are we? All right. So now we're at a position where our translational approach from taking microbes from from humans and putting them into germ-free mice don't seem to be working very well. So what are we going to do? Well, I have a very, very clever graduate student called Ellie Glennie, and her protege is right here in the audience, Willow. And basically, she came up with an idea. Now, she wanted to look at this phenomenon of caloric restriction in a much more controlled environment. So basically, there's many things that can happen to you when you severely reduce your nutrition, and one of these things is GI dysfunction. Now, I hope you can appreciate how clever I thought I was when I did this. Yeah, this is it's brilliant. Mirroring and mice, and there's a mirror, and everybody's gonna love it. And the first time I showed it, everybody's like, what are you talking about? Anyway, so we wanted to come up with a more controlled environment, and Ellie came up with this caloric restriction study. Now, there are caloric restriction studies where you reduce the available calories to a mouse by 20 to 30%. And what happens is those mice, and indeed humans, are so healthy. They live longer, they have better cognition, and that's like, I was like, so I'm like, Ellie, we can't do this study by reducing it 30%, it's beneficial. She goes, I know, I'm going to reduce it by 60%. And that is something that's going to mirror, acute starvation. And I was like, okay, well, let's have a look. We'll talk to our Aya cook and see if they let us. And they said no. And we said, oh, come on, please. And they said, okay. So we have to, we had to prove to them that these mice weren't in that much distress, and they're not. They're actually okay. And so what we do is we have a severe caloric restriction for 9 days, and then we look at the intestinal morphology, permeability, the microbiota, of course, and what Ellie's really interested in is the intestinal epithelium. So this is the most obvious graph I can show you. By restricting caloric intake by 60%, the mice have less food, okay? Okay, well done, okay. And also, predictably, mice lost weight very, very dramatically. Indeed, they lost it up to about 23%, which is fine because if we reach 30% of their initial body weight, we have to sacrifice these mice. But at 8 to 9 days, we're looking at 23 to 27% loss in body weight. And it's quite amazing. Gonadal fat is just a measure of fat in mice. In these calorically restricted mice, there's almost no gonadal fat left, so they're using up all of their fat stores. What's also very interesting, and there is a lot more data on this, but I'm just showing you like the highlights of what Ellie found, is that in the calorically restricted mice in the jejunum, the villi, which are your protrusions into your gut to absorb nutrients, are stunted. And this is very interesting because that could suggest that you have a lack of ability to absorb nutrients when you're in calorically restricted. So we also looked at permeability, and we gavage mice with Fitzy-dextran, I believe. Oh no, this was, yeah. And we found that the gut, although, so we're looking at truncated villus height, but we're also looking at increased permeability, okay? So we're getting towards this idea of a dysfunctional gut. And yes, the microbiota was different. So within about 9 days, there was enough change in the environment for microbes to be shifted away from what they were before they were deprived of nutrients. Now, we have collaborators who are really interested in intestinal stem cells. And this is a mouse called a SOX9 mouse. I won't go into too much detail, but this mouse has a fluorescent marker, EGFP, and depending on the degree of fluorescence, you can tell whether it's an active stem cell, a cycling stem cell. And what Ellie found was, in the calorically restricted mice, there was a bloom in intestinal stem cells. So that was kind of different than what I expected, because if you've got a dysfunctional gut, you would think you've got less stem cells, but here it seems that they're blooming. And then what we did was, we looked at the villus height in these calorically restricted mice, and then we also did it in germ-free mice. And we didn't see a significant decrease in villus height as we did in germ-free mice, suggesting that there is a role of the intestinal microbiota in this phenomenon. But as always, when we repeated, we didn't see the same thing again. But what we're thinking is of approaching this in a different manner by transplanting fecal microbiotas from SPF and calorically restricted mice into germ-free mice and seeing what effect they have on the villi. So in this model, Ellie has shown us that when you deprive yourself of nutrients, you change the microbiota, you change the function of the gut, and maybe your intestinal stem cells, which are responsible for producing the absorptive mass in your gut, are not functioning very well. And it kind of suggests that maybe we should be thinking about targeting the microbiota, which we know work in combination with intestinal stem cells, to recheck the ecosystem, so you're able to absorb the nutrients that you're giving to someone when you're clinically refeeding them. So I'm just, oh, okay. I'll just go over briefly what we're investigating right now. So this is a very interesting project to me. I've been working on it 4 years and I still like it. But what does it mean? And again, this goes to the hysteresis theory that we were hearing about yesterday. So what we want ultimately is to come up with alternative, safe and effective therapies for a disease that has a high mortality rate. So what does this mean? Are we going to give probiotics to AN patients? Well, why? We don't know what's going to happen. But we do know that the microbiota may not be quite what it should be upon exit from our clinics. So one idea is what Ajay was talking about yesterday, and he talked about yellow soup. And this was, you know, your fecal microbiota transplant. So we came with the idea that perhaps this would be a way of resetting the gut ecosystem in AN patients and, you know, perhaps helping them regain weight in a safer and effective manner. So really quickly, Ajay talked about this yesterday. If you have C. difficile infection, you just put in the healthy microbiota, it gets rid of the infection, and we're all good. But I don't think he talked about one specific study, which is the middle one there, where you can affect insulin resistance with this therapy. And we saw from Ajay's talk yesterday that there is definitely other diseases that can benefit from this technique or this method. So we work with a biobank, a stool biobank called MicroBiome. And basically, as Ajay said as well, they have a very, this particular company has a very high screening system, and they throw away 97% of all the fecal samples that they collect. It's parallel to a blood screening for transfusion. And you have your colonoscopy delivery, you have your upper GI delivery, or your pill delivery. And we had to apply to the FDA. I know this is one of the points that Ajay was trying to bring home yesterday, for an investigational new drug to use FMT in AN patients at UNC, and that's all you can do. It's a very, very specific requirement. You can't use it in any other hospital, you can't use it for anything else. It has to be UNC, it has to be anorexia nervosa patients. And we're basically going to take AN patients. They come in for 4 weeks for clinical refeeding, and we're going to give them an FMT once a week. And this is going to be a very small study. It's only going to have 10 patients in it. And our primary outcome is safety, to make sure this is not going to hurt anybody. And if so, then we can use this as a basis to move forward as a legitimate therapy for AN patients. And hold on. I just want to say that it's taken us a long time to get to this point, but they're actually doing this work in Europe. And there was one case report, it was an N of 1, but using, so you have to take it with a grain of salt, but the patient that they treated had a huge change in their weight gain by taking FMT with clinical refeeding. And it didn't seem that there was any consequences to the disease. Now, problem is, as Ajay said yesterday, we all have this drug. And this person in the top left is showing you on YouTube exactly how to do it. And if you look at it, it has 121,000 views. And I haven't watched it because it would make me angry. But I have read the comments on this YouTube channel, and oh my God, people are suggesting using this therapy for multiple things that have nothing to do with the intestinal microbiota. So this is a really important thing to consider, that people could do this themselves, and it's not a great idea. Now, the reason why it's not a great idea is that issue that Ajay talked about yesterday, where someone recently tried a clinical trial and transferred multiple drug-resistant organisms into susceptible individuals, and one of them died. Now, if you dig into the details of what happened here, is they didn't screen the donor, and they basically took the donor was working in a healthcare situation. They were working in a hospital. They're exposed to drug-resistant organisms all the time. So I don't know who approved that clinical trial, but it's really important that you think about the safety of patients as you move forward. Okay, so I'm ending a little bit early, but I just want to go through my funding. Thank you everybody for giving me money and keeping me doing everything that I can do. And I just want to also thank everybody that works with me, because basically I'm not allowed to go into the lab anymore, even though I want to. But I get in, they say, well, Ian, you're going to spend half an hour looking for a pipette, and then you're not going to know where the gloves are, and then you're not going to know where everything else is. So you're going to slow everything down. So I have a team or a village of individuals that are really the backbone behind all of the experiments I do, and I'm really grateful to work and collaborate with all these great people. And also we're working with different eating disorders institutes within the country. Now, this is what really, really scares me, because I wasn't born here. So that's definitely North Carolina. How do I do this? Colorado? Denver? Okay, I didn't know it was that far over. And this is Fargo. And I haven't visited there yet because I hear you have like 1 week where there's no snow. But all these guys work really, really hard. Actually, in particular, Denver have patients who really, really need our help. They come in to the hospital and they can't lift their head off the pillow. They're really, really sick individuals. And we have had individuals that we were trying to enroll in our study, but they died. So this is really, really important, okay? So with that, I'm going to thank everybody for listening to me. Okay, thanks.